All right. Well, welcome and congratulations on at least 50 years of graduation from the College of Science and Engineering. I'm delighted to give this Medallion lecture. And thinking about a topic that I want to engage you all with that might be timely. I wanted to focus on some of the research that we do in my lab, focusing on my favorite element, fluorine, which we're going to call the Maverick element today. I'll define that in a minute. And it's a type of element we hear about in the news a fair amount lately, sometimes called forever chemicals or PFAS materials, and it has a little bit of a dark side. But what I hope to convince you today about is that there's also a lot of promise in these types of fluorinated molecules. And if we can understand a little bit more about them fundamentally, we have an opportunity to both balance that peril with promising outcomes. And so I'm going to tell you about a couple stories focusing on aspects in human health and the environment. Although I started today with a slide here that I wanted to talk about myself, both as a researcher as well as an educator. I think the educational mission, particularly in the Department of Chemistry, but in CSE and UMN in general, is exceedingly important, as many fantastic educators. This is a slide of my family when I was young. This is me, and the reason why I show this here is to give you a feel of my background, which is I'm not a traditional first-generation student, although my father got his highest degree from Dormont High, so I'm from Pittsburgh. My mother actually did get a degree from college. She got a nursing degree from Carlow University in Pittsburgh, but she kind of did this through somewhat of a non-conventional way. She was actually in the convent. So my mother was a nurse, or not a nurse, she was a nun for 13 years. And you were in the convent, you could get a degree either in teaching or nursing. And she chose the nursing route, although the one aspect that was a little bit different is she was highly discouraged from actually talking to any of the students at Carlow University. And so her experience with college was drastically different of what you all may have experienced or what our students experience today. And so that has also shaped my experience of going to college, just learning how to college and being successful. Kind of bumbled along in my path as I've moved through the undergraduate education to higher education. That offers a unique perspective of talking to our students as they're also learning how to college, particularly our first-generation students. And I think that gives me a useful perspective so that all of our students can succeed, not just students that are more familiar with what higher education looks like. So I teach organic chemistry to our undergraduates here, and this is a cartoon a family member sent to me recently. And this is a dad in this first panel, and he has a kid that's crying, and he's saying, if you want something to cry about, I'll give you something to cry about. And the next panel, he hands the kid an organic chemistry textbook. And so this is what we're up against when we teach organic chemistry. This is a class that brings a lot of fear and apprehension and tears, sometimes thought to be a gatekeeper or a weeder course, particularly challenging for our students interested in a career in medicine. They approach this class, and many times with a lot of trepidation. And so we try, myself included, to really meet these students and engage these students and show that organic chemistry has a lot to give. In teaching them about the fundamental unit of a protein, an amide bond, an important organic chemistry functional group, and its stability. We note that the amide bond is found in proteins in your hair. So we'll have students come up, they'll cut my hair, and we'll kind of watch the hydrolysis rate in real time during the class. I'll freestyle rap about organic chemistry. We'll try these large collaborative quizzes where hundreds of students, instead of taking the quiz on their own, and the tears and fear that's associated with organic chemistry, will start to debate chemistry and talk about chemistry, challenge chemical ideas. And we hear laughter and enjoyment, and that's really helped their learning education. And so the real goal is not for the students to reach for the tissue, but to reach for the textbook. And that's where we're trying to meet our students, and that's where I hope you had your experience when you came through CSE and through our chemistry classrooms. But this isn't just me. So these 14 people, or 12 people here, are my colleagues right now in the chemistry department who are scholars in their respective research, but also eminent teachers, where all of them have been recognized as being distinguished teachers at the college or the university level. And I think this is the highest among all departments at the university, and I'm privileged to be a part of that. And particularly, David Blank, shown here, is also our Associate Dean for undergraduate programs. So we have very dedicated faculty in the education of our students. Here's just one more slide. This is just for some nostalgia. If you happen to be in Smith 100, this is where we would teach a lot of our general chemistry and organic chemistry classes. It was renovated in the mid-1980s, so it might look a little bit different for when you all were here. I took this during an organic chemistry class, and I don't know if you can see this very well, but this is a particular reaction over here. I'm just going to take a little bit of an aside for the moment. This is called the Diels-Alder reaction. I just wanted to comment on this because this is such an iconic reaction. And what I like to ask the students, and I'll ask you to think about, is have you ever been in a country where you didn't speak the language and perhaps you were lost? Well, here's a life-saving tip that if you are lost, all you have to do is shout the word Diels-Alder and look for who looks up. And if you find that person, you found a chemist, and they'll be willing to help you. Such an iconic reaction that people remember. The other thing, though, that you might remember is over in this panel over here in this blurry image is our periodic table. And during your time in general chemistry or organic chemistry, you might remember these periodic trends that we taught you all. As you go across the periodic table, the affinity for the electrons, called the electronegativity, increases. As you go down the periodic table, the size of the atomic radius gets larger. And we've talked about all these periodic trends. And one of the elements that sits in this kind of top right portion of the periodic table is fluorine. And that's the element that we're going to talk about today. So I call it a Maverick element, and if you haven't thought about the definition of Maverick besides John McCain or Tom Cruise, it's defined as a person who thinks and acts in an independent way, often differently than the expected or usual way, or in the words of our governor, it's just weird. And so that's what we think about fluorine. I mentioned that it is the most electronegative element. It wants the electrons the most out of all the elements on that table. If you make a surface of a fluorinated material, it is going to be extremely hydrophobic. You can watch your water bead up on it. Think about Scotchgard, where you've treated your linens. But also, it's extremely lipophobic. So it doesn't like fat. So it's not like oil and water. It has its own special phase called the fluorous phase. And if you look at this picture here, we actually have two layers. The top is a water-based solution, aqueous, it's just water. We have a goldfish swimming in it. And below it, this denser phase is a fluorous phase, which we actually have a mouse submerged in that. Why is that? Well, it actually was being investigated due to its really high affinity for oxygen as a blood substitute that you could administer to people injured on the battlefield in an emergency situation. Well, this mouse is actually breathing in the oxygen from that fluorous liquid. Weird, right? Unusual. Of all the elements on the periodic table, when fluorine bonds to carbon, it forms the strongest bond to carbon. In fact, we can't even burn fluorous materials. Weird, different. And that's led to this definition called a forever chemical that perhaps could be persistent in our environment, and that might have raised concerns for sustainability, and in fact it is, as these highly fluorinated materials are being regulated. Well, we embrace the weird in my group, and I'm going to tell you some stories about one technology we developed in my lab using organofluorine chemistry. One is a technique called protein observed fluorine, or PROF, NMR, where we're going to sneak in fluorinated building blocks of proteins called amino acids and use them as really interesting tags on protein surfaces to discover potential new medicines in this area of drug discovery. I'm going to talk about aspects of sustainability. When we were interested in the absence of fluorine in natural biological systems, we thought we could use this as an imaging agent, but we noticed these molecules were really highly fluorinated, and what happens when they get into our environment, and can we study them and develop alternatives? So we'll call that sustainable organofluorine chemistry. And a final small vignette at the end, I'll talk about medicinal chemistry, of how we took our drug discovery technology and worked in the field of chemical epigenetics, where we're trying to develop new therapies in the area of cancer and inflammation. So that's kind of where we're going to go today. After I graduated with a degree in chemistry from my undergraduate degree, I went to Switzerland as a Fulbright fellow. And one of the people I worked for was a guy named Jack Dunitz. He was really interested in some peculiar properties of fluorine. At the time, he penned a monograph called Organic Fluorine, Odd Man Out, because he recognized fluorine was also a bit weird. One of the things we teach undergraduates is high electronegative elements love to hydrogen bond. Lone pairs of electrons are with protons of other elements attached to electronegative elements. But fluorine, despite its high electronegativity, only forms weak hydrogen bonds, or hardly any hydrogen bonding at all. So despite that rule, it behaves differently. If you make a surface of a fluorinated material like Teflon, we know nothing likes to stick to Teflon. Water doesn't like to stick with it. Fat doesn't like to stick with it. It's a great degreasing surface. That's related to a property of low polarizability. Fluorine just really wants to hold on to those electrons and not share. Now, we could contrast that if we look in the Earth's crust. Fluorine is actually the 13th most abundant element. It's usually locked up in inorganic fluorine in the form of a mineral called fluorspar, or if you get the pure form, calcium fluoride. But it's absent from nature's recipes to make proteins or sugars or DNA. And so nature hasn't really encountered much fluorine in the organofluorine sense and doesn't know what to do with it. But we know what to do with it as chemists. This is kind of an old statistic up here. But over 20% of FDA-approved drugs and over 30% of agrochemicals, this is from the early 2000s, were noted to contain fluorine. Now, we clearly know these are beneficial compounds, so they can't be all bad, but they do have fluorine in them. And in fact, if you look at a more recent review, in 2018, basically half of all pharmaceuticals contain fluorine. Clear advantages, but they're becoming highly prevalent in our environment. Now, you can take the radioactive form of fluorine, F18, so you can generate this in a cyclotron, and you can label a sugar molecule of glucose, and look at sugar-addicted tumors in the body through a technique called positron emission tomography, or PET scans. In fact, over 90% of PET scans are all done using F18. We also think about imaging in the sense of magnetic resonance imaging, which we'll talk about today, or how we slip these into proteins using an in vitro technique, nuclear magnetic resonance spectroscopy, fluorine NMR, and I'll explain what this type of figure looks like. So it is odd, it is weird, but there are potential advantages of fluorinated molecules. Now, the peril associated with them is there's a class of compounds called poly and perfluoroalkyl substances, or PFAS. I don't think you can read the newspaper right now and not see an article about PFAS at least once a week. Because we're dealing with a forever chemical problem. These molecules are everywhere due to their beneficial properties. We can think about in our packaging materials for food. You know, think about your pizza box, or think about your microwave popcorn and the lining on the inside. These molecules are there, and then we're trying to phase them out. We can think about as a graduate from CSE, from chemical engineering. Robert Gore gave us expanded Teflon to help make Gore-Tex, a water-resistant, breathable material. In cosmetics, in certain cosmetics, over 50% of them contain PFAS. And if we think about the aqueous film-forming foams, or firefighting foams, AFFF, here you can see it kind of shooting out of this hose. This is leading to massive pollution with PFAS because they are so good at putting out fires. You can't burn them. They can form a film or a barrier over your fire that the volatile materials are not escaping, so you can't further ignite it. If we think about the strength of the carbon-fluorine bond, so here I show a dipole that the fluorine's pulling electrons to it. It is almost 30 kcals per mole more stable than a conventional hydrocarbon, shown here. If these molecules get into your body, so if they're persisting in the environment, they get into our food, we ingest it, or it gets in our drinking water, the average lifetime for these long-chain PFAS materials, such as perfluorooctanoic acid, PFOA, 2 to 8.5 years. Sometimes they get recycled in our body. Shorter chains, carbons of 4 or less, their lifetime's on the order of days, so that we do have some ways to mitigate this. Now the challenge from epidemiology, because they're lasting in our body so long, has been linking PFAS exposure to various cancers, as well as cardiovascular disease. Kind of the mechanism for cardiovascular disease perhaps has been looked into a little bit more in depth, looking at regulation of lipids and increased cholesterol, as serum proteins and fatty acid binding proteins can bind to these PFAS molecules. This also has effects in pregnancy, leading to hypertension or preeclampsia, low birth weight. And so this is why these PFAS materials are being regulated. The EPA has now set a limit for PFAS materials, set at 4 parts per trillion for 6 particular PFAS molecules, like PFOA on the last slide. This is kind of an interactive map. If you're interested, you can go to it. The dark blue circles, where there are a lot in Minnesota, are at the safe limit. In fact, you can type in your address over here and check out your regional area. Of course, there's also some hot spots that we have to think about ways to now break down and remove the PFAS material. And whether this is through activated carbon or reverse osmosis are important considerations in wastewater treatment plants. So what is PFAS? Well, the EPA has tried to work with this definition, defines it as a string of at least 2 carbons. One of them is fully fluorinated, the other one has at least 2 fluorines on it, or at least 1 fluorine atom on it. But neither of those carbons is going to have a hydrogen atom attached. So PFOA would fit that definition. But this molecule over here, fulvestrant, would also fit that definition. This is actually a life-saving medicine. This is a breast cancer therapy. It's a molecule that degrades the estrogen receptor called fulvestrant. In some more stringent definitions, a single fully fluorinated carbon, like this CF3, could be considered PFAS, and this is fluoxetine or Prozac. There are other medicines that are getting approved. You may have encountered this one. This one's on the commercials all the time. They got a kind of catchy jingle to it. But this molecule is Mibo. This is a molecule, it's a fluorous liquid that goes in your eye for treating dry eye disease. This is a very large market, not only in the US, but also in Asia. And so this is being produced on a commercial scale. Minnesota, folks may have also encountered TFM. This is a trifluoromethylnitrophenol. This is a molecule that we use about 30 to 50 tons per year. And this is used in our tributaries and streams that feed into the Great Lakes to take out a pest called the sea lamprey. which was a huge problem in the early 50s. And by treatment of the waters with this chemical that can selectively kill off the sea lamprey, we now see our trout returning, but we also have a lot of TFM in the environment. So there are benefits and challenges to PFAS, and how do we define them? I think one of the important things is that International Union of Pure and Applied Chemistry, the IUPAC, which if you ever took an organic chemistry class, those are the people that gave you the nomenclature rules that you may have liked or disliked. They have taken this challenge on as well. They convened a workshop in December 2024, and they're also looking at how can we better classify what are the definitions of PFAS, both for harmful PFAS as well as helpful PFAS, and how can we think about them. Okay, so how do we get involved? Well, I'm a biological chemist, and we started thinking about a spectroscopy tool to study proteins. So here's that amide bond I mentioned that's a building block of a protein, and it turns out you can label that amide bond with an NMR active nucleus, and at the most fundamental level, all the NMR active nuclei are going to do is respond to a magnetic field and tell you information about the local chemical environment. And so if you put these NMR active nuclei on every building block of a protein, cartooned out in the gray at the bottom, then you can get this two-dimensional NMR experiment where every one of these signals tells us about the chemical environment of each of those building blocks of the protein. This is called an HSQC NMR experiment. And so what my lab wanted to do is ask a question. And did we need all the amides, or could we have a more simplified experiment if we really wanted to understand interactions with our proteins to discover small molecule drugs? Could we have an image like the one on the right here, where these blue spheres represent the important sites of interactions where a drug molecule or a natural protein might bind? So to do that, we had to label a specific set of side chains on a protein. We do that with fluorine. So that's why we call it the protein observed fluorine NMR method. We love this method so much, actually, we got a social media hashtag, we got a license plate for this, so you really got to own it. But the reason why we do own it is, first of all, fluorine-19 is 100% abundant. So every atom you're going to get is going to give you signal in your experiment. The second thing is its sensitivity. It's the second most sensitive stable NMR active nucleus. So you can do these experiments quickly, quicker than these more complicated experiments over here on the left. The other thing that's important, and I'll give you one slide with some numbers on it, is fluorine is hypersensitive to those changes in chemical environment. So it's a great reporter on binding events, and it's also absent in biology. So we call it bioorthogonal. In fact, you can work, you can do these experiments in cells, you can do them in whole organisms. From environmental samples, you could even think about sampling sewage and just doing NMR in sewage and just looking at the fluorinated materials that are in there. So a pretty nice versatility with this nucleus. And this is what the data looks like. These spectra to me are simple and beautiful. These 5 signals here are telling about the chemical environment of 5 of the same amino acids which have a single fluorine tag on them. This molecule in the middle, called BRD4, we'll talk about that later, has 3 fluorinated amino acids called tryptophans. We see 3 signals very clearly. So when we're doing these experiments, we're dealing with biological molecules. So it needs to be really sensitive to detect them. And so we bought this Prodigy cryoprobe. This had quite good sensitivity. What that translated to, in about 4 minutes, we could work with 50 micromolar solution. So, 1,000-fold or 1,000,000-fold lower than your standard molar solution that you might deal with. So really dilute, tiny amounts for these 12 to 15 kilodalton proteins. Now at Minnesota, we have a better state-of-the-art probe. We can do these experiments in about a single minute. These are really great if you're looking at lots of small molecule binding events. We were trying to work with Eli Lilly to see if this would be of interest for their drug discovery pipeline. This is one set of data of some 3 molecules which we discovered which bound to a protein of interest with them. And we looked at the fluorine signal on the same building block and compared it to the hydrogen on that building block. So this is of a tryptophan amino acid. And the change in the signal response, which we call chemical shift change, is the blue bar for fluorine and the red bar for proton. What you can see if you don't understand anything from this is fluorine is significantly more responsive, 6 to 20-fold in this case. So this is what I've told you so far. Prof NMR is a really sensitive technique. Its sensitivity to chemical environment is really important. So this was my group in 2012. I would say, we could spread out all the signals in a one-dimensional experiment. We have signals of different shapes and sizes and can understand something about the chemical environment. But for magnetic resonance imaging, what you might encounter in a hospital setting, you don't care about lots of signals. You just care about one signal that you want to monitor. And that signal has to be really intense, just like my undergraduate Joe Manulik at the time. So tall and narrow is what you want for a signal for imaging. And so how do we overcome that responsiveness challenge of fluorine? That was something we had to wrestle with. Well, to give you a quick primer for MRI, so this is typically done for the proton nucleus. You will encounter protons in the most abundant source in water. In fact, we've got 2 of them in water, and our body's 80% water. So you can image water in your body and get resolution that's sub-millimeter. And MRI is not the most sensitive technique, but you can overcome this due to the high abundance of the water. And if you want to generate further contrast in your imaging, you can take advantage of a couple NMR relaxation properties called the longitudinal or the transverse, the T1 or the T2 relaxation properties. Even if you don't understand that, you may have encountered a contrast agent when you did go to the hospital. Might have gone in a magnet like this. This is at our Center for Magnetic Resonance and Research. It's a high-field clinical imager. This contrast agent uses a heavy metal, gadolinium. It coordinates water and can change the relaxation properties. It's a T1 agent, and that creates this natural contrast that we see in these images. Well, since fluorine's not found naturally in your body, could you have positive contrast using fluorinated molecules? Of course, we have far less of that than water, so we're going to need some tricks. So one trick is, could you make a highly symmetrical molecule, like these fluorous liquids, perfluorocrown ether? That'll give us 20 equivalent signals. We're tall and narrow like Joe. You get a nice intense signal. Now, if they're not magnetically equivalent, like perfluorodecalin, now you start spreading out those signals, you have ghosting artifacts, and your intensity goes down. So this is one way to do it, highly symmetrical molecules. The challenge with that kind of fluorous ball is it's not soluble in water. So you got to emulsify it to get it stable for imaging. And those emulsifications, those nanoparticles that you make, has stability challenges. So Amani Lee, a co-advised graduate student in my group with my colleague Christy Haynes, we looked at this problem. So again, these fluorous liquids, these dense phases, they dissolve high amounts of oxygen. And what we were interested is, could we develop an oxygen sensor using these fluorous liquids for MRI? Why do we want to do that? Well, with our colleague, Mike Garwood, he was really interested in implantable islets and a little Therasite device, which could be making insulin for diabetes patients. But if these cells grow and they run out of oxygen, then they're going to die and you're going to have to replace that device. So it would be helpful to understand how much oxygen you have. So one way to get around the stability problem, the fluorous liquids, is using the Haynes Labs ultramesoporous nanoparticles. Amani could load up the nanoparticles. Here you can see this dense core of perfluorous liquid. We would have these really strong images that we could get. And we could monitor an NMR property, the longitudinal relaxation time, which is responsive to oxygen concentration, to make various calibration curves to quantify oxygen levels. The challenge, though, from this plot on the right here is that it's also temperature sensitive. So Amani just didn't need an oxygen sensor, he needed a temperature sensor. So he went back to make a temperature sensor, and so he started looking at a molecule called perfluorotributylamine. That's a gold standard fluorous liquid. Looks like PFAS, though, and it's actually the most potent greenhouse gas, but it was the state-of-the-art temperature sensor. And so for temperature sensing for MRI, you actually need two signals that are going to change as a function of temperature. And through doing that, you can make your own calibration curve, like the one on the bottom. And so we wanted to redesign a new material that could be better than that. And so after a variety of computational and experimental approaches, Amani arrived at a rigid fluorous aromatic core and a flexible fluorinated tail. And he actually improved the responsiveness over PFTBA by about 55%. The challenge, though, was it was a solid, so we couldn't actually jam that into the nanoparticle. And we had some environmental concerns, because this long fluorinated tail is still a PFAS molecule, and so would this be highly persistent? So Xiaochen Li and the group took the challenge on to re-look at the aromatic core, as well as shortening the fluorous tails. And he ultimately arrived at this molecule called DD1. Why is DD1 interesting? Well, first of all, it is our most responsive. It's even more responsive than our first generation compounds. We can make it in a single step. So it's facile to do. And in fact, the building blocks are just hexafluorobenzene and a short thiol tail. And where this tail comes from is that synthesis of that breast cancer drug fulvestrant. And most importantly, it's a liquid, finally. So now we have this liquid that's ready to be encapsulated. And it also turns out it's not a forever chemical. It is partially degradable. One of the things that's helpful with this aromatic core, it's a chromophore that can absorb UVB light. So this is the light that your sunscreen is going to help prevent you from by absorbing that light. So with this material, it absorbs the light and then emits fluoride under simulated environmental conditions as a half-life of about 30 minutes. And so we've been studying the degradation of these compounds with a long-standing collaboration with my colleague Bill Arnold. And this represents now a way to think about better performing materials that might also be a bit more sustainable, which has got us very excited. So here's an example of some of the work we're doing with Bill. It turns out that not all carbon-fluorine bonds might be bad. And in fact, we have all different flavors of carbon-fluorine bonds. If we just look at the fluorinated pharmaceuticals and agrochemicals, here's a sampling of these fluorinated molecules that we've been studying and asking questions. How does each one of these types of molecules degrade? And what do they grade into? And so we can determine half-lives by HPLC, but we can also determine some of the identities of these fluorinated materials and the total fluorine count by fluorine NMR, and also couple that to a sensitive technique with mass spectrometry, and combine chemical shift prediction to also help in the identity. So this data over here isn't meant for you to understand it all, but this is just to show all the different types of fluorinated products that are forming upon exposure to environmental conditions. Here's a zoomed in picture, though. Here we look at fluoxetine or Prozac. So by total fluorine NMR, this is what we've started with. We can readily degrade it. This white bar is mostly fluoride. We have these two other species that are forming, and this third species, this black bar, we do know what that is, and that's trifluoroacetate. And so there's now a known mechanism of how this aryl CF3 breaks down into trifluoroacetate, an ultra-short chain perfluorinated carboxylic acid. This is that TFM molecule. If you remember, this is a molecule used to treat the sea lampreys. This also degrades lots of fluoride, but we also see, depending on the wavelength, different amounts of the black trifluoroacetate or trifluoroacetic acid forming. This is something to keep in consideration. TFA wasn't a regulated compound before, but it actually turns out in our waters, this is now beginning to be well over an order of magnitude, if not more, in concentration than other PFAS materials, either through getting into the environment from direct result of introducing TFA or as a degradation product of a lot of other fluorinated materials. And so this article here is starting to ask the question, do we need to start thinking about regulating and detecting this ultra-short chain perfluorinated carboxylic acid? Another interesting offshoot of our ability to develop techniques to detect fluorine really sensitively by NMR has been implications in environmental studies, where this is a collaboration with the USGS, with Bill Arnold's group. Kate Faber is the graduate student there working on the project. Here we were getting environmental samples from around the state at industrial sites or wastewater treatment plants, airports, agricultural sites, and determining the total amount of fluorine that was present through mass spectrometry and NMR. And mass spectrometry is far more sensitive, but to be quantitative, you got to know what you're looking for. It's called targeted mass spectrometry. Fluorine is agnostic. You don't have to know what you're looking for. And so we can more readily capture the total fluorine content, although it's less sensitive. And so what we found from this study, first of all, is how we could concentrate them. So we would take like a liter of environmental sample, would have to subject that to a mixed bed solid phase extraction, and then use an additional trick we've developed based on the literature from relaxation enhancement agents. So this is an iron ACAC. This allows us to more readily acquire NMR spectra and now be able to quantify the total fluorine amount from concentrated environmental samples. And where we can detect the amount of fluorine that's there, we actually get more total fluorine than mass spec levels, because there's additional products like these pharmaceuticals and the degradation products. So this is highlights an advantage, or at least a companion tool to mass spectrometry using quantitative fluorine NMR. All right, so we're going to finish up. I'm going to take a hard right and switch to the last talk about an area called epigenetics. And so one way to introduce you to epigenetics is you might have heard about the Hungerwinter or the Hunger Winter in the Netherlands in the north. And this happened during a blockade in World War II where the Dutch were sequestered off and being starved. They basically were eating grass and grinding up tulip bulbs, really undernourished until they could be liberated. Audrey Hepburn was one of the survivors of the Hungerwinter. And among the people that survived, they started looking at pregnant women in the Dutch famine birth cohort study. And what they found is the women afterwards were susceptible to obesity and cardiovascular disease, diabetes, multiple maladies. But that wasn't as surprising. What was surprising is their children were also susceptible to those diseases, and their children's children. So there's an epigenetic memory of this state, or what became known as an epigenetic memory. The environment was influencing the ultimate phenotype in presenting in these diseases, and it was persisting among generations. So one way you might think about your genes is the chromosome. We encounter this in early high school textbooks. This is how we package our genetic information. It actually turns out, though, that not all of our genes are accessible. And what epigenetics deals with comes from the word epi in the Greek, which means above our genome. And what it does is it dictates how our genes are expressed in a time-dependent manner, in a tissue-dependent manner that is heritable across generations. So how can we think about that? Well, if we think about the froglet to the frog, exactly the same DNA, so same genotype, but different phenotype. So genes are being regulated at different times. If we think about epigenetics in the context of disease, you can have a normal cell that all of a sudden takes on a disease phenotype, let's say a rapidly proliferating state, such as a cancer. There's a lot of interest and promise in epigenetic therapy of how we can reprogram these cells, not kill them off with toxic chemotherapeutics, but reprogram them to rewire the epigenetic memory back to the healthy state. And so we think about that as well in my lab. We study proteins that are involved with this. We use our fluorine NMR, and we also try to develop organic small molecules as potential therapies. For a group of folks who want to think about mechanism, one of the things I tell my organic chemistry students, the number one rule of organic chemistry is negative likes positive. So DNA is a polyanion. That's the purple DNA here. And these blue triangles are proteins called histone proteins. They tend to be highly positively charged, or what we call basic. So they have a lot of positive charge on them. They allow us to condense our DNA and package it in the chromosome and in our nucleus. And so our genes just aren't available to be transcribed. But chemistry can happen. You can neutralize that positive charge by acetylation. And when you neutralize that charge, the DNA now becomes more accessible. Our gene is ready to be expressed. And the proteins that we study are bromodomains. That's a fancy name of a protein that recognizes that acetylation event, recruits transcriptional machinery to carry out that process. So what happens if that gene is associated with disease? So here's a panel of cancer cells. This particular type of cancer, we know what causes it. We know what the driver is for this cancer. It's a particular gene called an oncogene called MYC. Now, MYC is dysregulated in about half of all cancers, and it's basically what I'll call the fastball of cancer. It tells the cells to rapidly keep growing. And the pharmaceutical industry has really struggled with trying to inhibit the function of MYC directly, MYC inhibitor. But a powerful approach outlined in 2010 was to use a small molecule inhibitor to stop MYC being expressed in the first place. And so this molecule, JQ1, inhibits a bromodomain called BRD4. It stops MYC expression as a potential cancer therapy. And so we and many other people have become interested in a protein called BRD4. This is an essential node in epigenetics. And so this is a picture of the machinery that has to come together. This is the bromodomain recognizing an acetylated bark. And when this machinery gets together, the outcome would be expression of genes like MYC, which can be any cancer target. Or we'll talk about other genes that affect inflammation. So JQ1 doesn't just inhibit BRD4, it inhibits a couple alphabet soups of related proteins, BRD2, 3, 4, and T. But there's a lot of excitement about it. Not only for that cancer I just showed, GSK showed that it could, analog of it could inhibit inflammation. It's been shown to be a possible therapy for prostate cancer. It was shown for blood cancers like acute myeloid leukemia. Shown as a potential therapy for a really aggressive tumor for childhood cancer, pediatric cancer called DIPG, where kids get a tumor in the brainstem, which is really inoperable. And the only therapy right now is radiation to try to shrink it. And even cardiovascular disease. So there's a lot of promise of diseases for inhibiting these bromodomains, which are part of a family called BET bromodomains. On these slides, I wouldn't pay attention as much to the numbers, but here's the 4 proteins. They actually have 2 bromodomains, D1 and D2, and the binding sites are just really similar. And in fact, molecules like JQ1 inhibit all of them. And the molecules that went into the clinic, that have since gone into the clinic, still inhibit all of these proteins, which are called pan-BET inhibitors. Now, despite the excitement about their efficacy, there's a lot of toxic side effects like thrombocytopenia and GI effects. And if you may have encountered thrombocytopenia, this is a platelet disorder. You may have heard of ITP. This was something I had when I was young. That's called idiopathic thrombocytopenia. It means we don't know the cause of this platelet disorder. But when I presented lots of bruises and blood blisters in my mouth, I've never seen my dad drive so fast to the hospital to get this treated out. Well, we know what the cause of this thrombocytopenia is, and it's targeting all of these BET proteins. So there has been some motivation. Perhaps you could target a subset of these bromodomains, like targeting the second domain. It actually turns out to be better tolerated, just fewer disease indications that you can use them for. So we have been motivated to develop more selective BET bromodomain inhibitors. We had a small lab starting out, and we had this quirky technique called Fluorine NMR, and we wanted to ask the question, could we use that to develop selective inhibitors? There's actually, even beyond those 8, 61 total human bromodomains. And could we target one specific bromodomain of that BRD4 protein as potential anti-cancer or anti-inflammatory therapies? This is my favorite data slide, so I always show it. It's a bit old now, but Andrew and the group recognized that these spectra were so simple that we were taking because BRD4 has 3 fluorinated amino acids that we were labeling. There are 3 tryptophans. BPTF, a related bromodomain, has a single tryptophan that we labeled, so 4 signals. And he said, well, why don't we just put them in the same test tube and see all 4 signals? And here we can see all 4 signals. And then he said, lots of people have inhibitors for bromodomains like BRD4. But really what you want is selectivity. So let's not look for potency. Let's just simply ask the question, what molecule would only affect a BRD4 signal, the tryptophan 81 signal, and leave the BPTF signal alone? And that's how we discovered this molecule 11B, this trisubstituted imidazole. He also asked which molecule would inhibit the BPTF signal. So it disappears, and now we have the 3 BRD4 signals alone. That's how he discovered this aryl urea called AU1, which we named after Andrew. This was the first inhibitor that was selective for BPTF over BRD4. It has moderate potency, but we've since been able to further generate next generation compounds and have a really established collaboration with St. Jude Children's Hospital looking at the role of these compounds in pediatric cancers, really focusing on neuroblastoma at the moment. But we're just going to talk about BRD4 today. And this is what happens when you do kind of these new selectivity experiments. You get surprises. This is an experiment called fluorescence polarization. All we're going to look for is just response. I'll just kind of walk you through the data. We took individual bromodomains, either the first one or the second one, and we subjected them to the experiment. When we looked at domain 2, it was a flat line, so no response with our inhibitors. But when we looked at domain 1, as expected, we saw inhibition. When we looked at a related bromodomain, BRDT, there was no response against the second domain. Only response against the first. This was unusual at the time. In fact, this was the most potent molecule that was selective for these first domains of the BET proteins. It's not that potent, though. This is a potency. It's a million-fold, lower concentration than molar, but not the concentration of a drug. But we could use that to ask simple questions in a multiple myeloma cancer cell. What happens to MYC? We're looking for the disappearance of the MYC protein, showing that it's not expressed. So JQ1 does that effectively. And our molecule, which we were calling HU10 at the time, also is effective. So now we needed a way to improve its affinity and understand why it's selective. And I'm going to skip over a lot of that development. And I'm going to show you how we arrived at our most potent inhibitor. So this is Huari. Huari was a graduate student in the lab. She was highly successful. Actually won the chemistry department's thesis award, was a runner-up at the University of Minnesota. A very talented graduate student. And this is one picture she got of her protein, where she appended this trifluoromethyl group in this aromatic ring, which was accessing a unique pocket on the protein that was distinct from any other inhibitor that had been reported. She also noted a particular water molecule through a technique called X-ray crystallography, and she's looking at something called a B factor, suggesting you could target this with your organic molecule. And she was showing we were way far away, and we weren't doing that. But we're chemists, and so we could redesign our molecule. And here's an example where now we can nicely target this water. We're slipping deep into this pocket. It's fitting what we call hand into glove. So this was a really nice indication of how we can engage these proteins. Companies like GSK are also using this strategy to target this water molecule. Now we're getting back to those response curves. The gray data is just reference data to non-selective molecules. So the second domains, now we completely leave them alone. We barely touch the first domains of every protein except for BRD4, which we nicely inhibit here. So now we have a really nice selective molecule. By calorimetry, it's 18 nanomolar. So now we're getting on the 1,000-fold more potent than micromolar. This is getting to concentration, so you might think about for drug-like molecules. So on in the group wanted to ask, we had a drug-like molecule. Let's go back and look at what happens with MYC, with this highly selective molecule. And so here's JQ1 again that gets rid of MYC. But what you can see over here, perhaps, is we're not getting rid of MYC. MYC's actually going up with these selective molecules. So we're not curing cancer. We may be causing cancer in this case. So for multiple myeloma, this might not be a good approach with these compounds and needs investigation. But liver disease is a little bit different when we're looking at inflammation. So my dad had a form of liver disease, non-alcoholic fatty liver disease. The only therapy that he had available to him was a liver transplant. And if you need a liver transplant, or know someone who's needed one, you know that you basically need to be end of life to be eligible for these scarce organs. So he was very excited when he found out that I was going to study liver disease. We got to meet Vijay Shah, is at the Mayo Clinic. And we started a collaboration not looking at non-alcoholic fatty liver disease, but alcoholic associated liver disease through focusing on alcoholic hepatitis caused from excessive alcohol consumption, where you can get a fatty liver, hepatitis, ultimately cirrhosis, and even liver cancer. So it's characterized by a high amount of inflammation, and really the only therapy or treatment for this that's effective is transplantation. So Vijay's lab had identified BRD4 being a crucial protein in this inflammatory process. It leads to expression of molecules like CCL2, which can recruit the innate immune system through macrophage recruitment, and then it's going to damage the liver. So the question was, would our BRD4 inhibitors not only inhibit this process, but be better tolerated? So we took a liver cell. These are liver sinusoidal endothelial cells, and with the non-selective BET inhibitors that inhibit all of those proteins, we could reduce the level of the gene CCL2. And in our compound that was selective, we could also do that, unlike the MYC case. So this was exciting. And that was in cells. But if we go actually in an organism, in a mouse, here we're looking at macrophage staining, those immune cells. You got a lot of brown when you can trigger acute liver injury with CCL4. See a lot of macrophages. You have a lot of less brown, less macrophage with your non-selective inhibitor, and similar with our selective inhibitor. So this was really exciting. As a quick aside, we also do look at these related compounds in cancer. We had to actually get rid of the fluorine and got rid of this piece of the molecule. And with my colleague Joe Topchevsky, we made this methyltriazole here. And this is now potent for BRD4. We do inhibit all of the BET proteins, but only half of the bromodomains. And when we go back to that multiple myeloma cell line, now we can potently inhibit MYC, whereas our selective compounds do not. So that was encouraging. And actually, I'm from Pittsburgh, and so I don't know if anybody knows who this is. This is D'Angelo Williams. He played for the Pittsburgh Steelers. He was a running back. And you can kind of see a little bit of pink in his hair, but it turns out a lot of his family members, the women on his side, contracted breast cancer. And he tried to be an advocate for breast cancer. He would wear pink cleats or pink shirts or pink in his hair, and he'd get fined by the NFL. But he kept doing it for advocacy. And he ultimately started a foundation. And so I always appreciated that and thought we would name our molecule after, in honor of him, DW34. And so we actually send our papers to his foundation. His wife turns out to be a biochemist, which is kind of cool. So we've taken DW34 as well as our BRD4 selective compounds. We're going back to liver disease, we're trying to make our molecules safer and more efficacious. This is showing molecule 3 is DW34. This is one of our BRD4 selective compounds. These are still working in vivo. This is looking at acute liver injury from taking acetaminophen. But we're also looking at other inflammatory effects. What was excited about this study by Cole and Nora from our group was the safety question. And so this is getting back to that thrombocytopenia, the platelet issue, that these non-selective molecules present in patient or treatment with these non-selective molecules leads to this process in patients in the clinic. So the lower the number, the more susceptible this assay predicts these patients will be to thrombocytopenia. So more selective inhibitors that target the second domain are much better tolerated, so a higher number. And we wanted to see what happens with our even more selective molecules. And so we looked at our BRD4 selective molecules and our DW34 molecule. And what we find is a second domain 2 selective molecule has a relatively high number as expected, but now one of our BRD4 selective molecules now is much better tolerated. And so we're really excited about the prospect of these molecules as a potential for safer therapy. So where are we going? There's a lot more organic chemistry to do. There's a lot of properties that you have to develop besides just potency. So we use these radar plots to visualize our data, thinking about their physical chemical properties, how we can formulate them, potential off-target toxicities, and we want to kind of fill out this pentagon in color, so showing we're addressing all of these properties. So we're still working on this. This is a work in progress. We just initiated a startup company. So I will disclose that as a potential conflict of interest, but we don't have any money yet. We haven't really started. But this has been as a collaboration with the Mayo Clinic and the University of Minnesota. We call it Bromothera. We're also very interested in asking the question, could these potentially be diagnostic imaging agents? And we would have to figure out how we put a radioactive 18F on any of these types of molecules. So those are some of the exciting things that are happening in the future. So just to summarize, hopefully what I've shown you is I've introduced some challenges of PFAS molecules and talked about perhaps how we could design more sustainable materials such as those fluorescent liquids for temperature sensing, thermometry applications. I've talked about how we're using sensitive fluorine NMR to detect the degradation of these processes. I've talked about how we could use our fluorine NMR method as a way to discover new medicines, or potential new medicines like Molecule 1 or DW34, and the development of those compounds, particularly for alcoholic hepatitis, is still going. These are the people that are in the lab now, and I've tried to highlight the people that did the work. These are the wonderful collaborators. All of the funding agencies, both statewide as well as nationally, that have helped supported this work. Thank you for listening to this talk. Congratulations on a milestone of 50 years since your graduation. And as a reminder of our education mission, we have a beautiful new teaching building that's coming this fall. They're actually turning the keys over to us this summer. In July, we're going to have a ceremony Would love to see you all there. So please do come back and also acknowledge first David Blank, which helped shepherd this project towards approval and getting generous contributions from the state in our bonding bill, and Christy Haynes, who is now our department head, that's been overseeing the construction. So we're really excited about this. Again, thank you for listening to this talk. And we hope to see you on campus soon.